Hi, hello friends, welcome back to my channel. You're watching Farm Pretty where learning is made simple, easy and fun. So in this video, I'm going to give you a detailed explanation about DNA and structure of DNA, what are the types of DNA, and you are going to detailedly learn about the DNA structure. So first, what is DNA? So DNA full form is deoxyribonucleic acid. So it is a hereditary material which is present in our body. So when a child is born, the child has the hereditary character of the mother, right? This is because of the DNA. So it is very important in our body. And when a child is born, for the child's information like hair color, eye color, and more information is just saved in the or collected in the DNA. So the DNA is mainly located in our cell nucleus. So while cell division or something else, this DNA also divide or form a daughter cells. So you will learn how it is dividing, how the functions are going on. So this process is just biggest process. So you will learn in this end of this video. So DNA is the most important thing that is usually found in the mitochondria, cell nucleus, in the cell nucleus mitochondria. So DNA is found by Watson and Crick. So DNA is present in both eukaryotes and prokaryotes. So just comment me your eukaryotes or prokaryotes. So DNA is also present in virus. So it may be double-stranded or monostranded. Also present in parasites. Also it's also present in the DNA. So DNA is a polymer of deoxyribonucleotides. So it is composed of monomeric units like DAMM, that is deoxyadenylate and deoxyguanylate, deoxycytosine and deoxythiolinate. So it is just named after the purines and pyramids. So also in eukaryotes, DNA is surrounded by nuclear membrane, whereas in prokaryotes it does not have nuclear membrane. So we know prokaryotes is single, that is non-nucleated. So there is no nuclear membrane around it. But in virus, there is a virus just has a protective layer that is called protein layer. So I have already told what DNA is found by Watson and Crick. These two members have found the DNA and given the term double helix model structure. So DNA is just described the name as DNA before many years ago, but after that slowly they have developed the technology and found that DNA is double helical, and it is just explained by these two members, that is Watson and Crick, that is double helix model. So DNA structure. So DNA is the right-handed double helix structure. So if you see a DNA, it just turn to right side, right? When you draw also, you just turn the DNA to right side. In the back side of this picture also you can see that it's turned to right side. So it is right-handed double helix, and it consists of 10 nucleotides pairs. Okay, 10 nucleotides pairs. Also, DNA consists of sugar, phosphate, and bases. The below picture is clearly explained the structure of DNA. So in this you can see a round structure, that is called phosphate group. The phosphate group only helps to attach each and every deoxyribose sugars, that is the DNA each other. And then the pentose shape is the pentose sugar. And rectangular shape, it may be any bases like adenine, guanine, thymine and whatever. So what are the bases? So the bases are mainly divided into two types, that is pyramids and purines. So to remember the purines element, just remember purine has Y. The same way thymine and cytosine also has Y. So the thymine and cytosine will always be with pyramids. Just remember like that. And then purines. Purines are adenine and guanine. So we know four bases: thymine, cytosine, adenine, and guanine. Thymine and cytosine belongs to pyramids, and then adenine, guanine remaining is belongs to purines. So the ring consists of that is pyramids consists of 6-membered ring only, but purines consists of 6-membered ring with the pentose sugar, that is 5-membered ring. So purines consists of both 6-membered ring, that is benzene and a pentose. Purines consists of only benzene. So this can be explained in the upcoming picture. Just listen to this DNA structure. There is a phosphate group, that this phosphate group attaches the each of the pentose sugar. So Actually, DNA consists of nucleotide and nucleosides. Nucleotide is the pentose sugar and phosphate group plus base. That is what nucleotide. Whereas nucleoside is the only pentose sugar and a base. Okay, so you remember this. We have a trick to learn that. So nucleoside consists of S, right, in the name spelling. So S, just remember S means sugar, right? S for sugar. Then sugar and base comes together in nucleoside. So just remember sugar and base are friends. So they just only travel everywhere. So nucleoside has the friends like sugar and base, whereas nucleotide has all of them as friends. So next is the base structure. So if you see this in this picture, you can just clearly see each and everything structure. So RNA is single stranded, right? So it consists of for thymine it consists of uracil. And then in DNA, adenine, guanine, thymine and cytosine, whereas in RNA, adenine, guanine, cytosine and uracil. So next we can learn the cell and features of Watson-Crick model of DNA. So the DNA is a right-handed double helix. It consists of two poly-deoxyribonucleotide chains twisted each other on a common axis. The two strands are parallel, that is anti-parallel. So one strand ran from 5' to 3' direction while the other ran from 3' to 5' direction. Just compare this with the two parallel adjacent routes carrying track in an opposite direction. And then the width of a double helix is 20 prime, that is 20 angstrom to nanometer. Each turn or pitch of the helix is 34 ampere with 10 base pairs. Each pair are placed at a distance of about 3.4 ampere. Each strand of DNA has a hyperphilic deoxyribose phosphate backbone, that is outside of the DNA, and then inside it has a hyperbolic basis, that is outside. Just remember, outside is hyperphilic. Philic means just sphere, right? So it's just sphere of water, whereas inside it has some water inside. So just remember like this. The two polynucleotide chains are not identical but comparability, that is complementary to each other due to its base pairing. The two strands are held together by a hydrogen bond formed by a complementary base pair, that is A and T. Adenine and thymine has two hydrogen bonds, that is double bond, whereas guanine and cytosine has three double bonds, that is three hydrogen bonds. And the hydrogen bonds are formed between a purines and pyrimidines only. The only base arrangement possible in DNA structure from spatial concentration is A to T, T to A, G to C and G to C. And then the complementary base pairing in DNA helix proves Charloff's rule. The genetic information series on one of the two strands known as template strand or sense strand. So genetic strand is collected at one side, that is called template strand or sense strand. The opposite is anti-sense strand. The double helix has a major groove and a minor groove. Along the polydiester backbone, protein interact with DNA at these grooves without interrupting the base pair and double helix. So below you can see a complementary chain, that is first chain it has ATACG, right? So you have 3A, 1T, 3C and 1G. And the opposite strand has just the same. So TA's opposite is T, right? So just anti-parallel. So next is the DNA is the double helical structure. So see this picture clearly. I have already told one side is parallel, another side is anti-parallel, right? So this is what is structure, actual structure. If you take a DNA and just make it straight, you will get this structure. So one side there is a phosphate, pentose sugar and A, C, G, T, A, G. So this is what in a straight position, right? Just see the opposite direction. The P is the opposite and T, everything is in opposite direction. That is tilted. So this is because this is anti-parallel. DNA stands for anti-parallel. So next we can see the conformation of DNA as double helix. So variation in the conformation of the nucleotides. Nucleotide means pentose sugar, base and phosphate. So these of the DNA is associated with the conformational variation of DNA. The double helical structure of DNA exists in at least 6 different forms, that is A to E and Z. So among this, B is form is the most important. So first one is A form. A form is also a right-handed helix. It contains 11 base pairs per turn. 11 base pairs per turn. This is the tilting of the base pairs by 20 degrees away from the central axis. So a helix is left-handed sense and short, and then it occurs in a dehydrated sample. If you take any sample of plant or something else, we just dehydrate that and then we just keep under the microscope. While observing that, you just see this A-form DNA structure. So next we can detail learn about the structural details. So A is fairly similar to B form. So it is slightly increased in the number of base pairs than B form. So B form has 10 base pairs, but A form has 11 base pairs. Just one number is increased. And then it has deep major groove and a shallow minor groove because it is getting after dehydrate, right? So major groove will be very deep, but the minor groove will be just slightly deep. So next we can discuss about B form. So I have already told B form is having 10 base pairs, that is spanning a distance of 3.4 nanometer, whereas A form has a base pair by 20 degrees away from the central axis. The width of the double helix is 2 nanometer. So the firstly we can identify this B form structure when you take it from our body. As it taken out of the body and just noted down, it has higher concentration of water. So next is the Z form, that is the Z form DNA. So Z form DNA is a left-handed helix and it contains 12 base pairs. It is just more than A and B form. So B form, just remember B form is 10 and then A has 11 and then Z has 12. So Z has the highest possibility. So one of the many possibilities is the Z DNA form, and it is most active double helical structure. It can be vivo, that is inside our body it is present. So in the upcoming picture you can detailing see the information clearly. B form is just, you can see that how clearly it was there, right? So it is just taken out of our body and just we noted down, and A, it is present in the dehydrated form, and then Z form, it is just present inside our body. And the last one is triple helix, that is this triple stranded DNA. Triple stranded DNA is formation maybe occurs due to additional hydrogen bonds between the bases. So there's a thymine can selectively form two halogen hydrogen bonds to the adenine, that is A to B pair to form AT. Likewise a protonated cytosine and more on. So triple helical structure is less stable than the double helical structure. So next we are going to learn about central dogma. So central dogma is about DNA, RNA, mRNA and the formation of these substances. So DNA is formed by transcription. DNA to RNA is formed by transcription, whereas RNA to protein synthesis is called translation. So this will be clearly and detailed explained in the next video. So I think you have understood this video clearly. So follow my channel for more videos related to pharm courses, and don't forget to subscribe my channel and also share this video to useful for many pharm students. So thank you for watching my channel. See you in my next video. Until then, bye. So don't forget to subscribe my channel.